Thank you for the invitation to attend this really exciting meeting. As you're here, we haven't had any applications for GM animals, so it's really useful to hear what's going on in the world. So I've been asked to speak about the public comment process in Australia for gene technology updates, and also to give an update on what's new at the OGTR. So first, I'll start by setting my context, which is the Australian Gene Technology Scheme. So as you're aware, the gene technology regulatory schemes across the world are all different. So I'll highlight some of the features of Australian regulation. I'll then briefly cover the transparent public consultation process that we use when we're administering the legislation, and then move on to consultation when the scheme is being changed. I'll also present some information from the Australian Food Regulator, because unfortunately they were unable to attend. So the Australian regulatory scheme consists of a Gene Technology Act and regulations. These came into effect in 2001, replacing a previous voluntary scheme. So we've been celebrating our 20th anniversary recently. So all work with GMOs requires authorization from the Gene Technology Regulator. And this includes work with any organism. So we regulate, microbes, plants, animals, insects, fish. We also regulate all the way through from work in labs right through to commercial release. We accredit organizations to work with GMOs. We also certify labs from physical containment 1 right up to physical containment 4, which are the high containment labs to do work with high-risk pathogens. And the regulation forms an integrative legislative framework with other regulators. So we work with product regulators, and I'll talk a bit more about that later. Now, unlike in many countries, we have a single decision maker, the Gene Technology Regulator. Now, she interacts with and gets advice from scientific experts, Ministry for Environment, and other regulatory agencies, but ultimately she makes the decision whether or not to approve work with GMOs. The assessment of risks from GMOs is made on the basis of scientific evidence. So it's a comparative assessment where the GMO is assessed against the non-GM comparator. And I think we had a bit of discussion about that yesterday after Eric's talk. So we would only look at risks from the GMO. Our processes are transparent and consultative. And as part of that, we have a public record of all the GMOs used in Australia. We also have monitoring and enforcement powers to ensure that the legislation is complied with. And they sit within the Office of the Gene Technology Regulator. So the object of the Gene Technology Act is to protect people and the environment by identifying risks posed by or as a result of gene technology, and by managing those risks through regulating work with GMOs. So to highlight here, we don't look at trade and marketing or socioeconomic effects. We are only looking at health and safety of people and the environment. We also only have to manage risks. We don't have to have no risk, and we only look at risks from gene technology. So the level of regulation depends on the risk. So for low risk activities, there is low oversight, and we focus our attention on higher risk activities. So work with certain GMOs on a list are exempt dealings if they're done in containment. For instance, in a lab or animal facility, and they don't need a formal authorization at all. So this would be work with, say, E. coli. We then have these notifiable low-risk dealings, and these are also done in containment, and they're overseen by an institutional biosafety committee at the organisation doing the work. The committee does have an independent member, so it's not completely the organisation. And they have to assess the work with GMOs, whether it fits the category, in line with the gene technology legislation, and then they notify their decision to the OGTR. So then there is risk, there's higher risk work for GMOs. which is either high-risk working containment or anything that's going to be released into the environment. So for a trial or for a commercial release. And this needs a license, which is issued by the regulator, and the assessment is done on a case-by-case basis. So for some examples, we have research work on GM animals done in containment, approved by the Institutional Biosafety Committee at the organisation. This would be common in most countries. Similarly, we have work with large animals. This is research work. These are some GM cows that are in a PC2 large grazing animal facility. Now, some of these are in buildings, but a number of them we affectionately call the PC2 paddock, because it is a field that has a double fence. It has locked gates. It's formally inspected. Those barriers are formally inspected every 3 months. There is signage and the animals have to be identifiable. But that's where it's been determined that the risk of a GM cow doing anything other than escaping is kind of low. We also have contained work with gene drives. This is research work, and this requires a license from the OGTR as it's considered higher risk due to the potential consequences if these organisms escaped. However, that's the extent of the work with GM animals in Australia. So the regulators issued 37 licenses for commercial release since the beginning of the scheme. As you can see, these are plants and also therapeutics, both for people or animals. We also have 129 trials since 2001. Again, these are plants, algae and therapeutics. All of these approvals require a science-based risk assessment involving extensive consultation. And this is where the transparency of the Gene Technology Act comes into play. So any applications for a trial or commercial release are publicly notified, and copies of those applications are available to anyone on request. We also then prepare a risk assessment and risk management plan. So we prepare that at the OGTR, and then that is sent out for public comment. It's notified on our website and it's sent to various subscribers. It's also sent to various government departments so that they can comment on our risk assessment. We then take those comments into account and the regulator will make a decision whether or not to issue a license. That is then also, her decision is publicly notified. The final risk assessment and risk management plan is also publicly available. And we also include a list of any submissions that we received and how they were addressed in the risk assessment and risk management plan. And this is sent back to anyone who makes a submission, and it's available on the OGTR website. We also have a lot of risk assessment material available, so our risk analysis framework, but also we produce biology documents. Now I'll switch topics and move from what we do every day to the recent reviews of the legislation that regulates GMOs. So the OGTR has two kinds of reviews. There's reviews of the gene technology regulations, and they're done by the OGTR. This is a technical review, and we can't change the policy settings of the legislation. The second type of review is a review of the whole scheme. Now, this occurs every 5 years, and this can change the policy. So this is where you can change the scope of what a GMO is. And there's also a review underway from Food Standards Australia, New Zealand that I'll talk about. So to provide, for context, so this is the definition of what a GMO is in the Gene Technology Act. So we have a process-based trigger. The process determines whether it's captured by the Gene Technology Act or not. As you can see, gene technology has a very broad definition to capture new technologies as they develop. And because they're broad, there is detail in the regulations which can declare something to be or not to be a GMO. So the regulations review that we did looked at the new techniques of gene editing to see if they produced GMOs. So I'll start by explaining the terminology that we use. So as you all know, you can use various site-directed nucleases to make a targeted double-stranded break. This can then be repaired by non-homologous end joining, which is a random process, and we call this SDN1. You can also use homology-directed recombination, which is guided by an added template. This can be a short template or a long template. So we call this SDN2 and SDN3. There is a very blurry line between SDN2 and SDN3. I don't think anyone's ever come up with a definition. So that is obviously a problem for legislation where you need something definite. Okay, so there was a discussion paper released in October 2016, 4 options of how these new technologies could be regulated, which are shown there. This paper was open for submissions for 2 months, and we got, for an Australian consultation, we got quite a large number of submissions. I realise this is very small compared to other countries. As I said, 126 direct, and there was a Friends of the Earth campaign from which we got another 600 submissions. This was, very useful to get the submissions. There was the direct submitters, about 1/3 from public and research organisations, also companies. So there was generally broad agreement that option 1, no amendments, was not appropriate, but there was a lot of disagreement about which of the other options were appropriate. Okay, but then finally, after a long government process of making legislation, these were the changes to the regulation. So organisms with the SDN1 modification, so where there is no template used to drive the repair, were considered not GMOs. However, if a template is used to drive the repair of the double-stranded break, then it does result in GMOs. Part of the reason for this decision was because under the regulations review, we can't change the policy settings. So this was looked at further under the review of the scheme. There was also, as part of this review, a decision that work with gene drive organisms would all require licensing, whereas previously contained work could be done under a organisation approval, and also clarifying that null segregants are not GMOs. And these amendments came into force in October 2019. So concurrent with this, there was a review of the whole gene technology scheme, which started in 2018. And this is run independently of the OGTR by a policy team. So they undertook consultation, I think 3 phases of consultation, and they came out with recommendations. So to maintain the process regulatory trigger, so we weren't going to go to a product trigger. It was still, if it's done using gene technology, it is regulated. They wanted to amend key definitions to clarify the scope in order to keep up with technical advances such as the new breeding technologies, and introducing risk tiering to facilitate flexibility so that there would be possibly other levels rather than just the notifiable low risk dealings and license, that possibly a permit could come into that category where you would not have to do a full assessment for things that the regulator is very familiar with. Also considering mechanisms where the regulations and the legislation could be updated more quickly. So I've put up 2 documents there that have been out. So there's been the issues paper gone out for consultation. The implementation is ongoing, and there's still further consultation expected on actually draft legislation, which is expected next year. Obviously, the pandemic has led to a lot of delays,
and the change of government has meant that it's hard to get drafting of a gene technology bill when the new government has other priorities. Okay, now just to go back a bit, I'm going to talk about the Food Standards Australia-New Zealand legislation, and I just wanted to put this slide up to remind you where they sit. So the OGTR is seen as a gap filler. We regulate work with live and viable GMOs, and we interface with all the other product regulatory agencies. So we avoid duplicating regulation where another agency has oversight. We align decision-making, and we talk regularly with the other agencies. So as an example, if you had a genetically modified cow, to import that cow or semen or embryos into Australia, you would need approval from the Department of Agriculture, Fisheries and Food for import, as well as from the OGTR. To have that cow in a field, you would have to have approval from the OGTR. And then for food safety, you would go to Food Standards Australia and New Zealand for an approval. Now, as I said, Lisa Kelly was unfortunately not able to be here, but she sent me these slides. So they have also started working on new breeding technologies in food. So this was initiated in February 2020, following a review by FSANZ, which found the current definition of GM food in the food standards codes were outdated and no longer fit for purpose. So late last year, FSANZ proposed a general approach to revising the definition, which included product-based exclusions for foods equivalent in risk to conventional products based on specific criteria. So standard transgenic products will still continue to be captured for pre-market assessment under this approach. So there were diverse views about the specific approach being proposed. So FSANZ has been analysing the submissions and taking on board the feedback received to inform the development of revised definitions. They've also undertaken consumer research in the form of a quantitative survey to supplement the information received through the submission process. And they're also developing additional communication material. So they intend to engage with a number of key stakeholders before finalising the proposed approach and preparing the revised definitions. And these proposed revised definitions will be released for public comment as part of a second call for submissions report, again in early 2023. So there's quite a large amount of work in this, and there's quite a large amount of material available on the FSANZ website, including earlier reports, the submissions from the consultation last year, consumer research and communication material. And they're planning more communication material to be released next month. And I'm sorry, that's all I know about this process. As I said, it's a sister agency. I was just going to have at the end a couple of slides on a community attitude survey that the OGTR has commissioned every year since 2015. So this question here was about the level of support for various science and technology developments. So as you can see, support has increased in 2021 for almost all kinds of GMOs. So support is highest for medicines and lowest for cloned animals, which is similar to the data that Alison showed yesterday for cloning. And this was another slide from the survey. Oops. So they asked whether genome editing will improve people's way of life, and 57% of people thought yes. But I think pretty much just as the survey came out, we also received this campaign letter. So, you know, which is obviously against genome editing. So, you know, are the public concerned? Obviously, some people are concerned, but it looks like the vast majority think it is a good thing. This survey is available in whole on the OGTR website. I will leave you with a photo of the staff at the OGTR. So this consists of scientific evaluators, monitoring and compliance staff, staff who work on the legislative reform, and also the people you need to run an office. So thank you very much for your attention.